coral bleaching is the failing of the coral tissues, and this happens through the loss of its algal symbionts or the photosynthetic pigments that the algal symbionts have. And bleaching is basically a stress response of the corals. It can have lots of causes, but the most well known is high temperature and high light. And that tends to happen in summer during very still weather events because there's very strong stratification in the water layer, so the top layer really warms up and a lot of light penetrates the water as well, so a lot of photo energy is hitting the algae and the tissues. We care about it because bleaching is really bad for the coral. The coral relies on the zooxanthellae for its food. It can receive up to 90 or 95% of its nutrition through photosynthesis by the algae. So once it loses the algae, it basically starts starving. Bleaching also causes the production of a lot of bad sort of chemical compounds. We call them reactive oxygen species, and they damage the tissues in the cells of the coral. So it's both a lack of food and damage to the coral tissues, and that can lead to, in the end, can lead to mortality of the coral, but it has a lot of detrimental effects. It can lead to reduced fecundity of the coral, so less reproduction, higher susceptibility to disease, and generally the corals become sick and they may die.
It's been an amazing experience. We've set off from Cairns, visited, I don't know, maybe a dozen or so reefs. First stop Green Island, Arlington. We then steamed all the way up to Tideman Cay, Wilkie Island inshore, then continued inshore to Knight Island, Curd Reef, Wallace, up the top, and then all the way out to the outer reef to Rain Island. Amazing place, just amazing, so clear, such beautiful corals, fish, everything. We continued then south to Sandbank number 7 and finally ending up here at Lizard Island on our way south. So the purpose of the trip was to a number of things. Firstly, to exchange a number of temperature loggers that we've got deployed around the place. I think we've done 16 temperature loggers all up. These are little units that record temperature and pressure for us, i.e. tides, and we have these going at least for the last 15 years or so. It's an annual thing, just changing them over. Secondly, we're interested in the connectivity of corals from one reef to the next, so we're sampling corals to look at their genetic markers and work out which populations are connected to which others. Also, adaptation, acclimatisation of corals to climate change. Again, genetic sampling to look at the different types of zooxanthellae that the corals have on board, and depending on the type, that tells us something about the acclimatisation state of the corals. We've got one more reef to visit on the way home, more reef, more temperature loggers, and we hope to be in Townsville after another 33 hours of steaming.
We're here at Lizard Island and we're doing a few things today. First of all, Ray will change his temperature loggers because he's following temperature over long periods of time. We will be collecting some whole coral colonies, hopefully colonies that have eggs, because we will spawn them at eggs. We will bring them back on the boat. Hopefully we'll also be collecting some samples for genetic analysis, both the genetics of the coral host, so we'll be studying connectivity on the Great Barrier Reef, but also diversity of the zooxanthellae that the corals harbour. And the zooxanthellae are important because we have found that they determine to a large extent the thermal tolerance of the coral, so we're interested to see how zooxanthellae types vary in space and time.